The goal of our work is to create a method that would be in understandable, accessible format allow experimenters to obtain visual physical characteristics of biochemical reactions. At the same time, we set ourselves the goals that obtained data could be directly compared with the data obtained in an in vitro experiment. Let's move on to a more detailed consideration of such an example. Consider the first example. The figure shows two graphs showing the results of the effect of mutations in the BAC peptide on binding to the BCL-XL protein. The graph on the left is from an experimental study. IC50 independently confirmed in competition binding assays using surface plasmon resonance. The graph on the right is obtained by numerical simulation. The y-axis indicates the value of the logarithm of the condition number for dimers of mutant and wild types. In this case, the value of log con W is directly proportional to the experimental value of dissociation constant. Thus, the values in the lower region of graph correspond to the more stable dimeric complexes, the stability of which decreases with increasing values of log con W. Thus, the results of numerical simulation are in good agreement with the results obtained for the same dimeric complexes by experimental methods. The main physical parameters that can be obtained by the software package are condition number logarithm, change in the measure of differential entropy, potential energy of electrostatic interaction between all involved amino acid residues of a dimer. The calculation of these values allows you to take into account the determination of affinity changes, but they are not equal to such experimental quantities as the dissociation constant and IC50. Now let's see how it is easy to use the intuitive interface of the program and get the graph data online. Let us proceed to the visual implementation of the introduction of a mutation into the BAC peptide upon interaction with the BCL-XL protein. Let's press the multi-parameter protein analysis button so that each mutation can be introduced in turn. Enter the name of the PDB file in the dialog box, which contains information of the three-dimensional structure of the BCL-XL protein. In the second dialog box, enter the name of the PDB file of the peptide, in which the replacement will be performed. There will be three replacements. In this case, the dialog box for the first interaction in our program is intended for the interaction of wild-type proteins, in relation to which we will analyze the change in the stability. And now let's move to the mutation input. We do not enter any substitution into the first vector, since that is BCL-XL protein. In the second vector, we enter the sequence number of the substitution, which corresponds to the number in the database. Q77 corresponds to the number 9. Next replacement is A85. It is serial number 17 in the PDB file. And the last mutation at number 10 in the PDB file. Press the button Standard Algorithm. Let's choose an orthogonal transformation and then press the button Standard Algorithm. The calculations are over. Let's see what graphs we got. The graph of dependence of LG con W on the serial number of the replacement is presented. Next, we have the same graph, but only in the form of dot representation. Thus, it will be more convenient to compare it with the experimental obtained graph below. A positive LG con W value is directly proportional to the dissociation constant. The upper graph demonstrates a decrease in the affinity of each other mutant form of the BAC peptide to the BCL-XL protein. Thus, the calculated data are in direct agreement with the experimental data obtained by the plasmon resonance method, and you can see the general conclusion from these two graphs.